Well, good morning, everyone. My name is Jason Franklin, and my presentation topic for you today is developing c-BERST to identify RNF2 complexes regulating GATA6 expression in PDAC. PDAC in of itself, PDAC is pancreatic ductal adenocarcinoma, and it is a deadly disease with an estimated 51,000 people dying from it in 2023. The 5-year survival rate for it is only about 12%, with that percentage decreasing the further the disease progresses. Ring finger protein 2 has been identified as a possible protein of interest for PDAC, playing a role in both metastasis and inflammation. Our group has previously identified RNF2 as a protein overexpressed in tumors from Black patients, where it may play a role in the racial health disparities of the disease. Our goal is to identify protein complexes at the GATA6 loci, a known master regulator of PDAC subtyping. By looking specifically at the RNF2 binding site within the loci, we can understand the mechanisms by which it inhibits the expression of GATA6. This process will be assessed via the DCAS9 Apex2 bioaccumulation at genomic elements by restricted spatial tagging, or c-BERST. c-BERST allows for the rapid, unbiased mapping of proteomes near defined genomic loci. So, before we really get into it, there is some background that should probably be established, such as cancer as a whole, pancreatic cancer and its health disparities, as well as current identification options, and then finally some explanation of the methods we use during this process. So the basic definition of cancer, according to the National Institute of Cancer, is cancer is a disease in which some of the body's cells grow uncontrollably and spread to other parts of the body. These cells can be any cell within the body, including gamete cells. Typically, a cell grows and divides as needed by the body, and when the cell grows old or becomes damaged, it undergoes apoptosis or programmed cell death, and then new cells take their place. When the system stops working and the cells begin to grow uncontrollably, the mass of fast-growing cells is then now called a tumor. Tumors can be both benign or non-cancers or malignant cancers. And there are 10 things that define cancer, what a cancer cell is. And I am going to go through all 10 of these just briefly here. And the first one that we're going to go through is the beta and growth suppressors. And cancer cells typically inhibit tumor suppressors, and there are factors within cells that prevent their growth. And one such growth suppressor is the RB protein. The RB protein integrates signals from within and out of the cell, and in response decides whether a cell should proceed with its growth and division cycle. Our cells have a delicate balance between growth factors and growth suppressors. So when the RB gene in the DNA is suppressed, this can lead to a cell's unchecked growth. On the other side of the coin, with sustaining proliferative signaling, cancer cells typically have mutations in the RAS proto-oncogene. And when it's turned on, it triggers a complex signaling cascade that introduces cell survival and growth signaling. So if the protein mutates, it upsets the balance once again and induces continuous replication and cell division. Both of these processes typically occur because there are mutations in the host cell genome. As we have seen with RB and GRAS, mutations can make them do one of two things: either make them stop working or make them work too well. Another phenomenon seen within the genome is that the repair mechanisms are suppressed, if not completely damaged beyond repair. If these repair mechanisms are suppressed, any subsequent mutations cannot be repaired, thus allowing the cancer to create an environment where it can best survive. As all cancers have some sort of mutation in their genome instability and mutation is known as an emerging hallmark. There's actually one other emerging hallmark, and the reason that these are called emerging hallmarks is because they often promote other of these hallmarks to actually appear. The other one is tumor promoting and inflammation. And the reason that this is an emerging hallmark is because inflammation of the tumor can contribute by supplying the bioactive molecules to the tumor's microenvironment, including growth factors, survival factors that limit cell death, pro-angiogenic factors, as well as factors that inhibit or induce invasion and metastasis. So because these are emerging, they're often well known. But there are two that are a lot newer coming to the table, and they are deregulating cellular energetics and avoiding immune destruction. Starting out with deregulating cellular energetics, a cell gets its energy through a process called glycolysis, and it's where glucose molecules are broken down and harvested to make ATP for the cell. And this process cannot occur without oxygen. And oxygen produces that last little bit of energy needed to finalize this process. And if oxygen is not present, a process known as lactic acid fermentation will occur. And the net ATP that it produces, glycolysis produces 32 ATP, while this process only produces 2 ATP. Normal cells only do this as a last resort when there's no oxygen available. And however, even in the presence, normal cells only do this as a last resort when no spare oxygen is available, because it is damaging to the cells and to our body, because it also produces lactic acid, which when built up in our muscles, it's why after a long workout or a long day, our muscles feel tired. Now, even in the presence of oxygen, cancer cells can reprogram their glucose metabolism to primarily use this process. And this has been termed aerobic glycolysis. And while it is counterintuitive, this allows for the fast production of ATP, for this 2 ATP, to give the tumor enough energy to grow effectively. The other characteristic that is actively being studied is avoiding immune destruction. And it has long been thought that the immune system does work in removing small tumors and singular cells when they are found in the body, but it can't remove the large tumors as they secrete an unknown suppressor. However, in recent cancerous mice models, the CD8 cytotoxic T cells and the CD4 Th1 helper cells have had deficiencies seen within the models. And it has also been discovered that the combined deficiency of these cell types has actually increased the possibility of cancer development within the mice models. These results indicate that the immune system's innate and adaptive responses contribute significantly to immune surveillance and thus tumor eradication. However, it is still unclear at this time what causes the deficiency in these cell types and what allows tumors to continue to evade the immune system. One of the more concerning aspects of cancer is that it can move throughout the body or metastasize. And there are 6 steps to metastasis shown here. And the process starts with invasion. In this step, the primary tumors are established and growing well within the body, and most cells in the tumor are expressing E-cadherin, which creates anchors for the cells to connect to one another. And when the tumor undergoes metastasis, it downregulates E-cadherin and upregulates N-cadherin, which essentially works the opposite as E-cadherin. The tumor cell morphology changes, which allows it to move through the epithelia of the primary tissue, where it moves actually to the blood vessel, where step 2 occurs, intravasation, where it simply enters the blood vessel. Once it's in the blood vessel, it enters step 3. And in the blood vessel, it follows the direction of blood flow in the body and moves to the next major organ. And this is why we can say that we can actually predict where a cancer will develop, is because the metastasizing cell follows blood flow, attaches to the major organ. Once it reaches the next major organ, step 4 occurs with extravasation, or exiting the blood vessel. Finally, in the metastasized cell, the secondary tissue begins to upregulate E-cadherin again, or step 5. And the cell then looks normal again in the body and will not be attacked. From here, one of 2 things can happen in the secondary tissue. The cell can stay in the area and not grow and enter a state of rest. Or the cell can grow in the secondary tissue and form a secondary tumor. One question remains though. How do the new tumors in the new area get the nutrients needed to grow? And the answer is angiogenesis. Angiogenesis is the creation of new blood vessels. And cancer cells induce this, or cancer cells induce this to ensure that nutrients is given to the tumors. In normal bodily functions, the process only occurs in the early stages of development in the embryo and uses the VEGF pathway to induce it. Cancer cells hijack this pathway and induce it anytime they need nutrients to the tumor. The tumor induces angiogenesis by producing VEGF. Unfortunately, in doing so, the newly formed blood vessels are very weak and often termed leaky. And this is why that many patients with cancer have to be careful when they become injured, because if these blood vessels were to rupture, very easily to do so, it could cause internal bleeding. Angiogenesis is actually one of the few hallmarks of cancer that can actually be seen as a physical symptom. The final two hallmarks of cancer that I'll talk about here are less symptoms and more of how cancer can last as long as it does. And they are resisting cell death and enabling replicative immortality. And when a cell becomes damaged or mutates beyond repair, apoptosis or programmed cell death can occur. Apoptosis can be induced with either intracellular or extracellular factors. And each pathway has a pro-apoptotic and anti-apoptotic factors acting in a delicate balance. The intracellular path for this, there are two main proteins, BACs and BAC. BAC is embedded into the mitochondrial membrane, and when it interacts with the BAC's protein, it induces the perforation of the mitochondrial membrane. This perforation causes the release of signaling proteins such as cytochrome C. The release of cytochrome C in turn activates a cascade of caspases that induce multiple cellular changes and induce apoptosis within the cell. Cancer cells can disrupt this pathway by downregulating BACs and BAC. They can also upregulate anti-apoptotic signals within the cell and short-circuit the extracellular apoptotic proteins. Without apoptosis, the longevity of the cancer cell is significantly increased. However, even without disrupting apoptotic factors, the cancer cell still has increased longevity due to how it affects telomeres. At the end of our DNA strands are repeating sections called telomeres. These telomeres help determine the cell age because after replication, these telomeres are often left in an overhang. And after replication, this overhang is cut off. After enough cutting off, there's no more telomeres, and at that point, if it continues to replicate, it will start to lose coding regions in the DNA. So cancer can achieve immortality by lengthening the telomeric regions of the DNA, and it does this by activating an enzyme called telomerase. Telomerase is usually only turned on during the early development stages as well, and when it's turned on, it does what it's supposed to do, lengthens these telomeres so that they never reach the state of normal. So the 10 hallmarks of cancer are what make a cancer cell unique, but cancer in of itself is also very broad. There are 200 types of cancer, and these types of cancer are typically named for the organs or tissues where they form, and there are 4 broad types: sarcomas, neuroectodermal, hematopoietic, and carcinomas. Sarcomas are cancer of the bone and soft tissues, and are one of the rarest cancers to form. Next, sarcomas, neuroectodermal cancers, are those that form in the brain and spinal cord. In fact, both of the types, sarcomas and neuroectodermal, only account for only about 5% of total cases. The next type of cancer would be the hematopoietic cancers. These are cancers of the blood and blood-forming cells. There are 3 types: leukemias, lymphomas, and myelomas. Leukemias are cancers of the circulating blood cells, and they are unique because they do not form solid tumors. In fact, they affect individual cells, and these individual cells can build up in the blood and bone marrow and block normal red blood cells from receiving nutrients. Next are the lymphomas, or cancers that begin in the lymphocytes, B, T, and natural killer cells. The abnormal cells gather in lymph nodes in the body and block the immune cells from doing their jobs effectively, and they do form solid tumors. Finally are myelomas, which are cancers of the plasma cells. They build up in the bone marrow and form tumors in the bones across the entire body. The last type of cancer is, with 90% of cases, is carcinomas. Carcinomas are cancers of the epithelial cells that cover the inside and outside surfaces of the body. Our focus is the adenocarcinomas, or cancers that form the epithelial cells that produce fluids or mucus. Most cancers of the breast, colon, or prostate are actually adenocarcinomas. So where does this all fit into what we work on? Well, pancreatic ductal adenocarcinoma is the most common type of pancreatic cancers. And it was difficult to diagnose. It is difficult to diagnose as many of the symptoms do not appear until later stages, and this makes treatments and prognoses a lot less than nothing. And PDAC forms when the exocrine cells in the pancreas grow out of control. And PDAC also has two unique subtypes: classical, which prevents cellular communication, and basal, which promotes cell growth and prevents cell death. There are many risk factors attributed to PDAC, such as obesity, diabetes, and tobacco use, but it can also be attributed to age and a person's ethnicity. This figure here shows the incidence rate for Black patients identifying as African American and African Caribbean. And you can see that any of the Black patients have a much higher incidence rate when compared to the other two races. This is also seen in these stages of diagnosis, where these patients have a much higher diagnosis, no matter the stage of the disease. So with this comes our goal for the product. Is there a way to identify PDAC more easily in Black patients identifying as African American or African Caribbean? Currently, the mechanism of identification of pancreatic cancer is using a CA19-9 blood test. CA19-9 is a carbohydrate antigen which acts as a cell surface glycoprotein complex. Under normal conditions, this protein is at low expression. However, it is highly expressed, and when it's highly expressed, it is usually a sign of pancreatic cancer. However, while this process is highly sensitive for detecting it, it is inefficient because high levels of CA19-9 can also indicate gallstones or cancers of the bile duct, colon, stomach, ovaries, or bladder. So the other way that we can identify or detecting these protein expressions is through antibody analysis, where an antibody is made to be anti-protein, whatever protein you may be looking for, on the Y end here. And on the other end, it is conjugated with a fluorescence marker. The red circle here is a protein that is expressed on the cellular surface. The antibody can then be detected through an imaging system or through flow cytometry, which analyzes fluorescence on each individual cell as they flow through an aperture. Through this process, our lab identified another possible protein of interest for the detection of PDAC. Our green finger protein 2, or RNF2, was one of the proteins of interest identified in our previous research. This volcano plot here kind of shows all the proteins that we looked at. And we specifically wanted to look at RNF2 because it plays a role in both metastasis and inflammation, as well as to be shown highly overexpressed in Black patient tumors. RNF2 has also been identified in urothelial carcinoma. However, its exact purpose is unknown. We know through previous research that RNF2 plays a role in oppressing several genes involved in development, differentiation, malignant transformation, and cell cycle. As we have identified this protein, we wanted to then look at the genome to see how this protein is regulated and what precisely this protein can regulate. Chip sequencing, or chromatin immunoprecipitation, is a technique for genome-wide profiling of DNA-binding proteins. This process allows for the precise mapping of protein-binding sites, and the process begins with the cells being treated with formaldehyde, allowing the proteins to become enriched. These cells are then sonicated or broken open with high-pitched sound frequencies, and an antibody specific to the protein of interest is used to immunoprecipitate the DNA-protein complex, in our case, the RNF2 protein. The antibody can then be detected through a fluorescence reader. Using ChIP-seq, our lab had identified GATA6 as a known master regulator of the PDAC subtyping, and as a significant loci for the PRC1 complex. This means that specific proteins are expressed from the GATA6 loci, which can determine the type of PDAC a patient can have, classical or basal. The GATA6 loci helps regulate differentiation, metabolism, and survival of resident peritoneal macrophages. The PRC1 complex, or polychrome repressive complexes, is a group of proteins that repress and help regulate the genes transcribed in the GATA6 loci. There are multiple variants of the PRC1 complex, each controlling different parts of the GATA6 loci. We wanted to identify which variant of the complex is localizing to the protein of interest on the loci. To do this, we need to be able to specifically look at the protein interacting on it, on the loci. A paper published in 2018 allowed us to do just that. It outlined a new method for rapid, unbiased mapping of protein with near defined genomic loci. And this is where c-BERST comes in. And this is actually a figure showing the basic process of it. And on the DNA here is a D-space Cas9. And this expands on the work of CRISPR, as we use a CRISPR-associated protein to act as an exonuclease and copy a part of the DNA just before our loci. When we say the protein is dead, it is because we have mutated to an inactive form. We do this because we want to use it as a starting point, not for it to cut the DNA, like a typical exonuclease would. Here, the dCas9 sits at the GATA6 loci, and then apex 2 sit attached to the dCas9, biotinylates all proteins that come in contact with the area. This biotinylation also adds a fluorescent marker, which we can see through the help of dots in shield 1 when added to the cell lines. Once we see red fluorescence in the cell line through a live cell imager, we can move into the next step of the protocol, cell lysis and streptavidin bead enrichment. Once we lyse the cells open, the proteins are then exposed to streptavidin beads, which can then be used to capture these biotin-labeled substrates. And these isolated proteins can then be detected through a mass spectroscopy system. The mass spectroscopy system that we would have used is LCMS, or liquid chromatography mass spectrometry. LCMS allows for the separation of the analytes in a fluid sample. And it works how a few microliters of a sample solution are injected into a flowing stream of solvent. The liquid sample is then continuously pumped through a column, usually filled with silica particles and a neutral buffer. Then, depending on the protein's chemical composition or physical properties, they are separated and pulled through the column. At the bottom of the column is a detection plate, and based on the mass or other properties, the proteins will hit the plate at different times. A connected computer can then detect this difference in times, and we can determine the masses of the proteins. And then cross-referencing with databases of proteins, we can determine what they are. So before we even can get to mass spec, we have to be able to add all these proteins that we're wanting in here into the cells. To do this, we need to code the fluctuating neutrons. And we use this by we code these proteins through the use of plasmids. Plasmids are circular parts of DNA that are found in bacteria, and we can isolate and engineer them in the lab to do what we need them to do. Once we introduce them to E. coli bacterial cells in a transformation protocol where the E. coli cells are heat shocked, the shocked membranes are then opening, the plasmid we introduce can then enter the cell and be replicated at mass. The cells are then plated onto nutrient auger ampicillin plates and left to grow at 37 degrees Celsius at 5% CO2. The c-BERST protocol requires two plasmids, one that encodes dCas9 apex 2 protein here, and another that encodes the guide RNA. Basically where that guides this protein where to go onto the loci. The apex 2 plasmid, shown here, is an engineered peroxidase that functions as an electron microscopy tag and a promiscuous labeling enzyme for live cell proteomics. There are a few genes to note here. The dCas9 gene, which codes for the main protein, the apex2, which acts for the biotinylating agent, ampicillin resistance gene, which allows us to grow on these highly specific plates, and finally, the M-cherry gene. The M-cherry gene codes for a protein that, when it interacts with DOX and shield 1, gives us that red fluorescence that we are looking for. The other plasmid we used was SGTLO. SGTLO is the guide RNA that can be programmed to lead the dCas9 to the loci of interest. And along with our inserted gene for the GATA6 loci, there are a couple of other genes. Again, the ampicillin resistance gene that we use to put onto these plates for growth. And the other one is this MTAG BFP2. This MTAG codes for a blue fluorescent protein that will be expressed in 293 T cells, which can then be detected through our imaging system. So in the case that we get a blue fluorescence and a red fluorescence, we know that both plasmids are in the cell and expressing their proteins, meaning we can move through to the next step of the protocol. Unfortunately, this was not the case regarding our experiment, as we did not get fluorescence in the 293 T cell lines, and there were other complications with the plasmids. The most glaring issue we faced was when digesting our isolated plasmids from E. coli, the HTTO was cut with the NCO1 restriction digest enzyme. And there should have been two very clear bands, one at 3.4 kilodaltons and the other at 5.2. What we did see was three bands, one at 9.1, 8.0, and 4.7. Even if we were looking for three bands, the top band should have been seen at 8.6, as that would have meant that there was still, that the enzyme used did not cut all the plasmid, and there was still undigested plasmid within our sample. Because of this, we were led to believe that we were actually sent the wrong plasmids from the company we ordered them from. However, upon sequencing through genome mapping, we found that the identity of the plasmids was actually correct, but there was possible contamination. The contamination was not only with another plasmid that the company had on hand, but also with our E. coli cells that we used. The other issue we faced was that even with pure and high concentrations of plasmid DNA, there was no fluorescence 48 hours post-transfection into the cell lines. Because of this lack of fluorescence, we cannot move forward with this protocol at this time. At this point, the plan is to continue to troubleshoot these plasmids so we have no contamination with other plasmids. We will also continue to transfect or introduce pure plasmids at high concentration until a successful transfection into 293FP cell lines can occur. Once that is successful, transfections into other cell lines such as ASPC1s, BXPC3s, and MIA-pocket2 cell lines would occur. These cell lines are specifically created as PDAC cell lines so that we can identify what is going on at the cellular level. The other thing that we can focus on is this protocol very much focuses on PDAC from a proteomic standpoint, or the protein-protein interactions within the cell. We can also look at this project from the genomic side. This would allow us to specifically look at the DNA and RNA interactions and see if there are any specificities within the cell. This would work because we can cross-reference the cell's purity with RNA and DNA sequencing. We can use the RNA sequencing to look at and find long non-coding RNAs, or LNCRs. LNCRs can modulate chromatin structure and function, as well as the transcription of neighboring and distant genes. By looking at LNCR, we can determine if there's anything specific or unique to PDAC that would be worth looking into. We would then look at the DNA sequencing to try and be able to find if there are any GOTA6 or GOTA6-AS1 chromatin interactions. AS1 is the gene that promotes cell proliferation as well as metastasis. If there are any interactions between the two, it could tell us if specific PDAC subtypes exhibit more of one hallmark than another. So in conclusion here, pancreatic ductal adenocarcinoma is a deadly form of cancer. It is so dangerous because it is challenging to detect until symptoms appear. The current detection option works, but it is inefficient as it also detects other GI issues. PDAC is also unique as there are significant incidence disparities within the African American and African Caribbean races when compared to the white and Latino Hispanic races. This created our interest in the disease as we wanted to see what the proteomic differences there were in the different tumors. We found that RNF2 is overexpressed in these patients, which could play a role in the health disparities. We have found that RNF2 is regulated by the GOTA6 loci, which is transcribed by the PRC1 complex. PRC1 complex translates different versions of RNF2 depending on where it interacts with the GOTA6 loci. We then found a new protocol, c-BERST, that allowed us to map the proteomes at defined genomic loci. c-BERST had two fluctuating components, the FX2 plasmid and the SGTO plasmid, both of which code for proteins for this protocol. The majority of active research was spent troubleshooting issues that arose with these plasmids. However, while we are still perfecting the plasmids to continue with this protocol, we have other directions such as RNA and DNA sequencing. And finally here, I would like to say that research is never a sure thing, as new discoveries and new ideas will continue to shape not only how we look at cancer, but the world. And with that, I will happily take any questions.
I have a question. Why did you, or were you assigned pancreatic cancer? Is there something that you're interested in? Is it within your family? Like, how did you get interested in that?
The reason that I was put in with this project was actually through an internship that I had.
Okay.
It was through the IMBRI program, and one thing that it allows us to do is allows us to do research at campus at UNMC or Creighton over the summer. and and I just kind of chose, and we're given like all the faculty that are in the program and kind of able to choose names. And I don't remember my exact reasoning, but I think I really like what project my mentor was working on at the time. And I was kind of happy to bring it back here once I was done.
Awesome, okay. Nice.
Jason, what was your method for transfecting?
For transfecting into the 293 cells,
Or the E. coli, whichever one you said, because you said it didn't work, you had no fluorescence, so it looks as if your transfection methods weren't working.
that I'm not entirely sure of the transfection
I
methods.
didn't know if you did a heat shock or if you did a...
Heat shock for the E. coli.
Okay.
Into the 293 cells, I do not know what reagent we used.
So you weren't getting success.
We were getting success at getting the plasma into the E. coli, but not the plasma into the F250 cells. So
So the 293.